in the Division of Cardiology, Department of Medicine at UCLA. It is my great honor to introduce to you Dr. Bob Lefkowitz from Duke University, who will be speaking at our lecture today. Dr. Lefkowitz is a highly accomplished scientist who has made so many fundamental contributions to the field of G-protein-coupled receptors. He's not only a leader in the scientific field, but also a great mentor to many generations of scientists. He's also a Nobel laureate in chemistry for his groundbreaking research on G-protein-coupled receptors. Dr. Lefkowitz was born and raised in New York. He went to Bronx Science for high school education, which is known to has produced the most Nobel laureates in science of any secondary school in the whole world, including 7 Nobel laureates in physics and 1 Nobel laureate in chemistry, which is Dr. Lefkowitz. After high school, Dr. Lefkowitz obtained his bachelor's degree and a medical degree from Columbia University and spent 2 years as an intern and assistant resident at the Columbia Presbyterian Medical Center. After that, he joined an NIH research program from 1968 to 1970 to pursue his research interest, which, as described by Dr. Goldstein and Dr. Brown in A Perspective in Science in 2012, is considered to be a golden era in the history of NIH research training, with 9 and now 10 Nobel laureates who graduated from this program, with discoveries from a wide range of scientific fields, from LDL receptors to oncogenes to nitric oxide signaling and G-protein and G-protein-coupled receptors. These physician scientists in this program are called yellow berets. Shown in the picture are 3 Nobel laureates: Dr. Michael Brown, Dr. Bob Lefkowitz, and Dr. Harold Varmus. As you can see, they all share a similar position in this group picture, third from the right on each row. You could consider this a lucky position if you want to take a group picture next time. After a brief stay at Mass General Hospital, Dr. Bob Lefkowitz went to Duke University to tackle the most important and the most challenging question in the field of G-protein signaling, and identified and cloned the first beta-adrenergic receptor gene. For his groundbreaking discoveries, he has earned many awards, including the 2007 National Medal of Science, and shared the 2012 Nobel Prize in Chemistry with Dr. Brian Kobiaka, a former cardiology fellow who was trained in his laboratory at Duke University. Besides his many contributions to the scientific field, Dr. Lefkowitz is also an outstanding mentor to many and role model to many trainees, both in and outside his laboratory. I can attest to that from my own research experience as a research fellow at Duke University. One of my most memorable and enjoyable moments during my training at Duke was the joint weekly lab meeting with Dr. Lefkowitz. Dr. Lefkowitz is truly inspirational and serves as an enthusiastic support for trainees. I would like to share with our trainees a famous quote from Dr. Lefkowitz: "Science is a 99% failure, and that's an optimistic view." Without further ado, please join me in welcoming Dr. Lefkowitz to our CTSI Distinguished Speaker Lecture Series today. Thank you, Dr. Lefkowitz.
Well, thank you very much indeed, Dr. Kai, for that warm and very nice introduction. So today I'll be speaking to you about G-protein coupled receptors, a subject which I've devoted the entirety of my 55-year research career to. So to begin, I think most of you must be aware of this G-protein coupled receptor family. It is by far the largest, most versatile, and the most ubiquitous of the several families of plasma membrane receptors. In fact, these receptors regulate virtually every physiological process in humans, including several of our sensory abilities, such as taste, smell, and vision. Most importantly, from the standpoint of clinical medicine, members of this receptor family are the commonest target of therapeutic drugs approved by the FDA. Now, when I first began working on these receptors more than 50 years ago, there was no consensus that they even existed. In fact, the prevailing intellectual climate is summarized on this slide from Earl Sutherland's Nobel lecture in 1971. He focused on the enzyme adenylate cyclase, whose discovery had won him the Nobel Prize that year, which of course leads to the generation of cyclic AMP, the first discovered so-called second messenger. Notice that in his slide, which as I say was the prevailing concept at the time, there was no such thing as an independent receptor, nor had G proteins been discovered. So it was felt that hormones and drugs like adrenaline and glucagon, which could activate cyclic AMP levels, did so by binding directly to this enzyme and allosterically stimulating its activity. By contrast, today, decades later, we have a much more complicated understanding of how these systems work, featuring receptors, G proteins, and a variety of other proteins that we'll be talking about today. So what I'd like to do in my lecture this afternoon is tell you how we got from here to here over the past half century, hanging much of the story on some of my own work. So let's get started. Back when I opened my lab 50 years ago in 1973 at Duke, there was no agreement that there was even such a thing as receptors as independent biochemical entities. Moreover, there was no way to study them. So during the first phase of my work, I concentrated on developing a suite of technologies which would allow us to study the receptors and then to hopefully isolate and characterize them. And the first such technique was radioligand binding. And so we were successful in developing radioligand binding techniques to study each of the several types of adrenergic receptor, the so-called beta and alpha adrenergic receptors. We also developed affinity chromatography resins, shown on the left part of this slide, in which we covalently attached beta or alpha adrenergic receptor antagonists to Sephiros beads, to solid supports. And then after solubilizing our radioligand binding sites from membranes, which were presumably the receptors, we could isolate them on such affinity chromatography columns. We were also able to covalently label the putative isolated receptors with photoaffinity probes that we also developed over the years individually for each of the adrenergic receptor subtypes. And so after 15 years of difficult and dedicated work, as shown on the right panel, we were able to isolate each of the then known 4 subtypes of adrenergic receptor, alpha 1, alpha 2, beta 1, and beta 2. 3 of them are shown on this single slide, each one labeled with a covalent photoaffinity probe, radioactive, or with its orthoprotein. You can see that each of these putative receptors was a glycoprotein with a molecular weight of about 60,000 Daltons. Each of these isolated proteins bound a variety of drugs, ligands, with just the right specificity and stereospecificity that one would anticipate if they were the true receptors. Some skepticism persisted, however, because it was said that, well, okay, you know that these proteins bind ligands and drugs the way they should, but can they do anything? Can they convey sensitivity to adrenaline on an adenylate cyclase system? And so the next phase of the work in the early 80s was to reconstitute these putative beta adrenergic receptors. Initially, we did this in cells, as shown in the bottom panel, using erythrocytes from Xenophus laevis, the African clawed toad. Why? Well, because most cells, mammalian cells, have beta-2 adrenergic receptors, we needed to find a cell which didn't. And through trial and error, we found this cell, which had adenylate cyclase and the just discovered at the time G proteins, and even binding sites for prostaglandins, but no binding sites for adrenaline, no beta receptors. So we took our putative receptors, reconstituted in phospholipid vesicles, fused those vesicles with the membranes of the xenopus cell using polyethylene glycol, and lo and behold, the cells now acquired responsiveness to beta adrenergic drugs like adrenaline. Within a year, shown in the upper right panel, we were able to reconstitute the entire system in collaboration with the late Eva Neer and Lutz Birnbaumer, our purified receptor, Birnbaumer's G protein, and Eva Neer's isolated enzyme adenylate cyclase. And so we could show that these three proteins alone reconstituted a fully functioning system in which adrenaline could stimulate the adenylate cyclase. So we knew for a fact we had the true beta adrenergic receptor. With purified, validated receptor in hand, we cleaved it with cyanogen bromide. We came up with 5 isolated peptides, and after several very difficult years, succeeded in cloning the cDNA and gene for the beta-2 adrenergic receptor, and learned at that point of its 7 transmembrane spanning domain arrangement. Architecture is shown here, which was supported by 7 stretches of hydrophobic amino acids of appropriate length, and connected by intra and extracellular loops. And most remarkably, and quite surprising, shown in blue circles, residues in the transmembrane domains which were identical to those in the visual signaling protein rhodopsin. And so in that moment, we realized that rhodopsin and the beta adrenergic receptor were actually related cousins, if you will, signaling molecules, and that they were the first 2 founding members of what would become the huge family of G protein coupled receptors. Over the next decade or more, many other laboratories would jump into the field. I should point out that in addition to the beta receptor, as shown on the bottom panel, within a year, we were able to clone the alpha-2 adrenergic receptor, again, based on sequence we derived from purified protein. Over the next few years, we would purify 4 different receptors, adrenergic receptors, clone their genes, as well as ones for serotonin. And very quickly, we had cloned almost a dozen G protein coupled receptors. And so others using those sequences and using homology techniques were over the next few years able to fill out this huge family of what became almost 1,000 different G protein coupled receptors. But, you know, virtually nobody again ever cloned one of these receptors based on having purified it to get sequences to develop probes for the cloning. Instead, they used a variety of homology techniques, PCR, etc., based on our published sequences. And so always felt very good about the first 15 years of very difficult work developing the tools that allowed us to purify the first 4 G protein coupled receptors. And now, of course, the family is known to be huge. I told you about 1,000 receptors. There are probably as many as 300 olfactory receptors alone. But the GPCRs as a family constitute the largest family of drug targets. About 1/3 of all FDA-approved drugs, that's 700 drugs, are ones which target these G protein coupled receptors, anything from beta blockers, alpha blockers, angiotensin receptor blockers, and then these new semaglutide type drugs, which everybody's raving about, and on and on and on. So the basic science work many years ago laid the basis for the development of so many hundreds of drugs. Now, right from the beginning, back 50 years ago, in addition to trying to isolate and characterize and prove the existence of the receptors, I was also fascinated by this phenomenon, which is called desensitization. We didn't discover this phenomenon. It had been known for many, many years, and it always intrigued me. Desensitization refers to this almost universal process in cells that if you stimulate them with a GPCR ligand like isoproterenol or epinephrine, the response, shown here as cyclic AMP, rapidly wanes over time despite the continued presence of that agonist. This just shows you that the same is true for angiotensin. It's true for basically all G protein coupled receptors. I think the reason that this always fascinated me so was that it's a perfect example of homeostasis, which is arguably the most pervasive and important of all physiological principles, namely that cells have multiple mechanisms when they are perturbed for returning to their original state. So we were always interested, whenever we would develop a new technique for studying the receptors, to turn it over to the desensitization problem to see if we could get any clues to the mechanism. And the first one really came about 1980, when we found that if we photoaffinity labeled receptors, beta receptors, from cells which had been desensitized by prior exposure to adrenaline, that their mobility of those receptors on SDS gels changed. So in the far right lane is the labeled receptor from control cells, but on the far left lane is the receptor from the desensitized cells. And you can see that it shows a somewhat retarded mobility in the SDS gel, which had at that time already been described as a property of phosphorylated proteins, especially membrane proteins, apparently due to their altered binding of the SDS detergent. Well, with that clue, we took cells, we labeled them with inorganic radioactively labeled phosphate, and as shown in the bottom, we were very quickly able to show that desensitization in this Turkey erythrocyte model system was associated with phosphorylation of the receptor. So over the next few years, and this work was going on in the 80s, a very talented graduate student, Jeff Benovic, was able to initially identify the novel protein kinase responsible for this phosphorylation. We initially called it the beta adrenergic receptor kinase, or BARK. He was then able to purify it from bovine brain and clone its cDNA, showing that it was the first or founding member of a new family of cyclic AMP protein kinases. Contemporaneous with this work, people working in the visual signaling field were studying a protein they called rhodopsin kinase, which phosphorylated rhodopsin. We were struck by the fact that the most salient characteristic of these two protein kinases, BARK and rhodopsin kinase, was that each phosphorylated their receptor substrate in a completely stimulus-dependent way. Only the activated beta receptor or the light-bleached rhodopsin could be phosphorylated. So we reasoned that maybe they were related protein kinases. So we were able to obtain some purified rhodopsin kinase from Chris Palczewski, and in short order, Wolfgang Lorenz in the lab was able to clone its cDNA, and so we were able to demonstrate that, in fact, they were very closely related in primary sequence. Over the next 5 years or so, we were able to isolate and clone a number of other related protein kinases, which by then we were calling G protein coupled receptor kinases for obvious reasons. And then in 2003, collaboratively with John Tesman's laboratory, we reported the first crystal structure as shown here. And you can see this shows what is now called GRK2, that is the renamed BARK, consisting of 3 domains, a highly conserved central catalytic kinase domain shown in yellow, 2 flanking domains which are more diverse, RH and PH domains, and it is bound to G beta gamma. G beta gamma interacts with 2 members of the GRK family, namely GRKs 2 and 3, which were previously known as BARK 1 and 2, in order to help them translocate from the cytosol to the plasma membrane. So today we know that there are 7 GRKs. GRKs 1 and 7 are limited in their expression to the retina. Only GRKs 2, 3, 5, and 6 are universally expressed, and between them, they're able to phosphorylate, as best one can tell, virtually all known GPCRs. But that was not the end of the story. Something is missing. Benevik, when he was purifying bark, GRK2, prior to cloning it, found to his distress, our distress, that as he purified the enzyme, which he was assaying by its ability to phosphorylate the beta receptor reconstituted in phospholipid vesicles, as he purified it to higher and higher specific activity, it was losing its ability to actually desensitize the receptor, which he was assaying by reconstituting G protein together with the receptor, and then looking at the ability of adrenaline or isoproterenol to stimulate the GTPase activity of the G protein. So we're getting purer and purer kinase, more and more phosphorylation ability, but less and less ability to desensitize. Just at this time, Hermann Kuhn, the late Hermann Kuhn, reported that an abundant retinal protein called 48K protein, or S antigen, was able to somehow work with rhodopsin kinase to deactivate rhodopsin. And the protein was accordingly renamed arrestin because it arrested signaling. Well, I called him and obtained some of his 48K protein to try it out in our system. And sure enough, Benevik was able to show that when added to our highly purified GRK2 kinase preparations, it seemed to restore the ability of the kinase to desensitize the receptor in our system. Well, our good fortune continued because almost simultaneously, Shinohara at the NIH reported the cloning of what was still being called S antigen, 48K protein, i.e. arrestin. He reported its cloning. By this time, we were beginning to think that the protein that we might be losing during our purifications might be not only functionally analogous with retinal arrestin, but that it might be actually structurally related to it, homologous with it. And so we obtained Shinohara's clone, and with low stringency screening, Martin Losey in the lab, a postdoc, was able to pull out a 70% identical protein, which we called beta arrestin 1. And then using beta arrestin 1 as a probe, a year or two later, a tramadol in the lab pulled out beta arrestin 2. So now we had three validated, closely structurally related forms of arrestin. Visual arrestin, beta arrestin 2. We then able to carry out this experiment shown in the right panel. In the upper part, we have a system of rhodopsin in vesicles phosphorylated with rhodopsin kinase. In the bottom panel is the beta-2 receptor in vesicles phosphorylated with BARC or GRK2. Now we're adding individually all three of these arrestin molecules and looking at the ability to decrease signaling. That's to say, an in vitro model system of desensitization. As you can see in the upper panel, at the visual rhodopsin system, the arrestin was very potent, but the two forms of beta arrestin were very weak. Conversely, for the beta receptor, the two forms of beta arrestin were very potent, and the visual arrestin was very weak. This was an important finding because it suggested that not only did members of the GPCR family share a universal structural architectural arrangement, but they also shared a universal regulatory mechanism consisting of a two-step process in which receptors were phosphorylated by a GRK only when they were activated, followed by the binding of beta arrestin to the phosphorylated receptor to lead to desensitization by sterically blocking G protein interaction with the receptor. Today, we know there are four forms of arrestin, as shown here. The nomenclature is a bit muddied because visual arrestin is known as arrestin 1, and beta arrestin 1 and 2 are also known as arrestin 2 and 3. The structure is shown here, and I'll talk more about the structure of the arrestins in just a couple of minutes. And so where we were by the early 90s, after about 20 or 25 years of work was understanding that we had a large family of 7 membrane spanning domain receptors, which could signal through G proteins, which could be phosphorylated when in their active conformation by GRKs, leading to the binding of beta-arrestin to the phosphorylated receptor, which was generally phosphorylated on its C-terminal tail, leading to desensitization by steric blockade. Over the next, I would say 10 years, there were multiple rapidly moving developments, many of them in our laboratory. For example, it was found by Jeff Benovic and Mark Carone, 2 trainees now working independently in their own labs, that beta-arrestins were clathrin adapters, which promoted the almost universal process of GPCR internalization. And we found, beginning in the late 90s, that much to our surprise, the molecules that we had discovered as the desensitizers of signaling were actually signal transducers in their own right, so that the beta-arrestins, in addition to desensitizing G protein signaling, could also lead to signaling in their own right. Initially, we showed this with things like SARC and MAP kinases, but then the number of processes or signaling pathways that were transduced through beta-arrestin grew and grew and grew until now there are many, many dozens, maybe even hundreds of such pathways. And then perhaps even more surprising still was that we found that certain ligands, certain agonists or stimulators could stimulate signaling down one pathway, say G protein, or the other, beta-arrestin, not both, which, as we'll talk about, initially presented quite a conundrum. We referred to such ligands which had disproportionate ability to stimulate one of these signaling pathways rather than the other. We refer to them as biased agonists. I don't have time to go through the mechanisms by which beta-arrestins transduce GPCR signaling. That's a whole lecture in its own right. But suffice it to say, there are at least 2 mechanisms which work in concert. The first, shown on the left, and which was the first discovered, was that the beta-arrestins act as scaffolds, which bind together several proteins that normally work in a sequential fashion, such as the members of the RAF, MEK, ERK, MAP kinase pathway. But in addition, they also serve as direct allosteric activators of their targets. And this has been shown with ERK and SARC and RAF. So we have both scaffolding and allosteric activation as concerted mechanisms for beta-arrestins transducing signals. But I want to talk now about biased agonism because that has major implications for drug development in the years ahead. So biased agonists are ligands which stabilize a particular active conformation of a receptor, thus stimulating some responses but not others. 7 transmembrane receptor ligands, for example, can be biased toward a particular G protein or beta-arrestin. Mutated receptors can also be biased. So classically, we taught that there are only 2 conformations of a receptor: inactive, usually denoted as R, and active, denoted as R star. And the idea was that agonists were those ligands which preferentially bound to R star, the active form, thus pulling the equilibrium in that direction. In such a simple schema, there's only one active form of the receptor, and all signaling occurs through that active form. But the notion of biased agents is completely incompatible with that if you can have ligands which can stimulate some responses and not others. And for that, we need to postulate there must be a multiplicity of active states, at minimum 2 for simplicity, or I should say oversimplicity, shown here. Now, there are significant therapeutic implications of that. Let me give you just 2 indications. Consider the mu-opioid receptor, which mediates the antinociceptive or pain-relieving effects of morphine. As shown on the left, these receptors, like all GPCRs, can activate and signal through both G proteins and beta-arrestins. But it's been shown that the beneficial analgesic effect of opiates are mediated through G proteins, GI, whereas many of the adverse effects, such as constipation, respiratory depression, and intolerance, are mediated through beta-arrestins. So what that means is, if you go over to the right side, if you had a G-biased agonist, which was able to stimulate G proteins, but not beta-arrestins, you should preserve the pain-relieving effects, but ameliorate some of the adverse side effects like constipation, respiratory depression, etc. In fact, one such agent is now FDA approved, and others are under a development. But you should bear in mind that the concept is somewhat controversial, and other factors may be at play. But here's an example where potentially a G-biased ligand would be desired. But one can also conceive of the opposite case. Consider the angiotensin and angiotensin receptor blockers as ARBs. ARBs, as you know, are amongst the most useful drugs in cardiovascular medicine. They do this because they're able to block, if you look over on the left, angiotensin can stimulate some adverse effects because it's a potent vasoconstrictor, can lead to hypertension, increased work in the heart, increased angina, etc. But beta-arrestins also signal downstream of angiotensin receptor and have been shown to have beneficial effects, such as mild inotropic effects and anti-apoptotic effects. So in this case, if you had, instead of a general ARB, and all the ARBs that we use in medicine are unbiased, or balanced ligands, which equally block G protein signaling, which is good, that's why they're useful, but they also block potentially useful beta-arrestin signaling. So if you had a beta-arrestin biased ligand, that might have additional therapeutic benefit. This has been validated in animal models, where in several animal models of heart failure, these agents are particularly effective. And various companies are trying to develop such Berestin-biased ligands for the angiotensin receptor. So in recent years, my lab has been particularly interested in trying to understand the biophysical basis of this biased agonism, which, as I explained to you, requires that there must be multiple active conformations, some of which may lead to all forms of signaling, some of which may lead to Berestin signaling, and some of which may lead to G protein signaling. So in order to understand this concept of how biased agonism might be generated, let me define some terms. So one potential mechanism of bias would be what we refer to as ligand or efficacy bias. Here, the differential signaling is due to ligand-specific receptor conformations. That is to say, each ligand-receptor transducer ternary complex is unique. Another mechanism is so-called system bias. This would refer to situations where there is differential signaling due to unequal pathway coupling. And this relates to the fact that each cellular environment is unique. For example, if you had a situation where the concentrations of G protein and beta-arrestin were very discrepant, and one was many, many-fold higher than the other, that alone might lead to bias, even though there was no bias at the receptor level. So let me at this point define a couple of terms. By molecular efficacy, I refer to the fact that the allosteric effect that a ligand exerts on the interaction of a receptor with a signal transduction protein, namely T. It describes essentially the ability for a ligand to assemble a ligand-receptor-transducer ternary complex. And so the question we wish to address is, can biased agonism originate from transducer-specific molecular efficacies? Now, our attempts to answer this question took me back to some work that I and Andrew de Leon had done together some 45 years ago, when we first developed the radioligand binding techniques. And what we had found, initially somewhat perplexingly at the time, is shown here. In the upper left panel, we found that if we did simple radioligand displacement curves with an antagonist, like alpranolol, we got steep, uniphasic Michaelian curves. But, as shown on the right, if we did the same experiment with an agonist, like epinephrine or isoproterenol, we got shallow curves, as shown on the leftward of the two curves there. We got shallow curves, which could be decomposed into two components, a high and a low affinity binding component. And remarkably, if one added a guanine nucleotide like GTP to the incubation, then the shallow curve was shifted to the right and steepened. And in fact, instead of having high and low affinity components, it now had only a single low affinity component, matching the lower of the two shown here. We could then calculate a ratio of KL, the low affinity, to the high, which in this case was about 100. Remarkably, we found that if we took a series of agonists of varying intrinsic activity, and by intrinsic activity, I mean the maximum ability to stimulate the adenylate cyclase system, where we're setting a full agonist like epinephrine or isoproterenol at 1. If we then use several partial agonists, and we make a plot of their intrinsic activity versus this KL over KH ratio, there was an excellent correlation. So we came up with the so-called ternary complex model to explain this behavior. What we postulated was that there were two forms of the receptor bound to agonist. The low affinity complex was AR, an initial low affinity complex of the agonist with the receptor. But then, in the case of an agonist, but not an antagonist, the agonist receptor complex could then combine with another protein, which we initially called X, to form an ARX high affinity ternary complex. The ability of an agonist to drive this second reaction, the formation of the ternary complex, is essentially a measure of its molecular efficacy. And that is reflected, of course, then by the ratio of these two affinity constants. While we were doing this work, Gilman discovered the G proteins. And since GTP converted high to low, it seemed obvious at that point that X was in fact the G protein. And this then, of course, explained why we had this wonderful relationship. 20 years after we published the ternary complex model for explaining agonist receptor G protein interactions, Benovich's group published very analogous findings with beta-arrestin, showing that the agonist receptor complex could also combine with beta-arrestin to form high affinity complexes and thus shift agonist binding curves progressively to the left. So with that background, we decided that we wanted to investigate whether transducer-specific molecular efficacies could explain physiological bias. And we decided to do this initially using the angiotensin receptor as our model. And the reason we chose that receptor was because over a period of years, we had developed a whole suite of analogs of angiotensin with varying degrees of bias. So angiotensin itself, as you can see in the upper green row, is an octapeptide. It has no bias. It activates its G protein, GQ, and beta-arrestin both robustly. But we had 4 peptides varying in 1 or 2 residues from angiotensin, which were Berestin biased. These are essentially loss of function mutations, which have lost the ability to activate GQ, but can activate Berestin. Then we have 2 shown down here in purple, which are GQ biased and which are gain-of-function mutants, retaining Berestin bias, but they're superactivators of the G protein. Okay, so how are we going to test this? Well, again, we decided to use the conceptual framework for understanding ternary complex models, which I've just explained to you. The idea was we made 2 types of fusion proteins attaching either G-alpha Q or beta-arrestin 2 to the carboxy terminus of the angiotensin receptor. We then carried out the simple concentration response curves, looking at the ability of each of these 8 or 9 ligands to displace radioligand from the receptor. In each case, comparing the untethered, unmodified angiotensin receptor with these 2, okay? What we anticipated was that to the extent that an agonist was able to promote the coupling of the receptor to G or Berestin. To that extent, we would see a leftward shift in the curve due to the ability of that agonist to stabilize some fraction of the receptor population in a high affinity form. And this ratio, again, of the low to high affinity would be a measure of its molecular efficacy, which we're here calling alpha T, or alpha transducer. And what we found, and published several years ago, was very much in keeping with that. For balanced ligands like angiotensin, there was a fairly equal shift in these curves, regardless of whether we use the G or Berestin fusion compared to the unfused receptor. If we had a Berestin-biased ligand, and again, all these biases had been determined in our lab by cellular experiments. For Berestin-biased ligands, they shifted the Berestin curves, but not the G, vice versa for the G-biased. And of course, antagonists led to no shift whatsoever in any of the constructs. Now, since these shifts, KL over KH, represent the molecular efficacy, we could then compare these values for each ligand. And in particular, we did this simply by just taking the log of those alpha values, those molecular efficacies, and subtracting them, okay? So here in this graph, we're looking at the log of the molecular efficacy of alpha GQ. Again, that's KL over KH. Same for Berestin. If we subtract the two logs and make a plot, we see the following. Now remember, log 10 is 1. So a value of 1 means there's a 10-fold better molecular efficacy at G than at Berestin, okay? And you can see that the values run anywhere from +1 for the G-biased ligands to -1 for the Berestin-biased ligands. So in other words, we have a lovely graded series of ligands with molecular efficacies ranging and bias ranging over 100-fold range. Okay, so now we have a very secure set of reagents to use. So how are we going to finally get at what might be different about the conformations of biased ligands, stabilized, the extent to which biased ligands can stabilize different conformations? And to get at that, a couple of years ago, we formed a collaboration with Wayne Hubbell's lab here at UCLA. And he is a pioneer in a biophysical technique called double electron resonance spectroscopy. This is an electron paramagnetic resonance technique in which one makes use of unpaired electrons in probes like nitroxide probes, which are covalently inserted into the molecule of interest. So in our case, we're particularly interested in looking at what's going on conformationally at the inner surface of the receptor, because that's, of course, where transducers are being bound. So just to give you an example, we know from crystal structures that the single largest conformational rearrangement when a GPCR is activated is an outward movement of transmembrane 6, as shown here. So consider if we covalently insert a nitroxide probe on the inner surface of TM6, and then another one on a much less mobile site, transmembrane 1, and then make Deer measurements. What it's able to do is to give us angstrom resolution distance measurements for the distance between the two nitroxide probes, and then probability distributions of the population of the receptor, which conform to each conformation or each distance. So if you look at the little insert here, in gray are the data that are obtained with the APO receptor with no ligand on it. And as you can see, there seem to be two populations of receptor with TM1, TM6 distances of about 32 and 35. But if we add angiotensin, you can see that about 75% of the receptors shift to a much longer distance of about 45 angstrom, which sort of makes sense. Okay, so by trial and error, difficult work, Laura Wingler in the lab was able to find 6 suitable sites for labeling on the inner surface of the receptor. To combine these, and we need 2 probes to make these measurements, combine these in 10 different combinations, and then make measurements in the Hubble lab with the 10 different angiotensin analogs and blockers that we were just talking about. So what did we find? Well, the first point is that there weren't just two conformations, inactive and active. There were, at minimum, 4 conformations. This is determined by putting all of the data I just described to you together and using a trilateration procedure to pick out the minimum number of conformations which could explain all that data. And that turns out to be 4. There may well be more. Now, in the diagrams I'm about to show you, the colored circles represent the average positioning of each of the probes, okay, in relation relative to one another in each of the conformations we discerned, superimposed on a ribbon diagram of the receptor for reference. The ribbon diagram in all cases is the inactive receptor. It's not the particular conformation being delineated with the colored circles. Okay. The other thing that we're able to get from the analysis, as I said, is the population of the receptor in each conformation with each ligand. So what did we find? Well, the first conformation shown here we call closed, because as you can see, the positions of the of these probes, that is to say that the inner surfaces of these transmembrane helices are in a very closed position, which you can see conforms very, very closely to where they would be in the known inactive crystal structure. You can see that this largely constricts the inner surface of the receptor and basically occludes the binding site. And since this would correspond to the inactive receptor. If we go over here in deep blue, you can see that the closed conformation is the predominant one when there's no ligand on the receptor, and it's enhanced even further when angiotensin receptor blockers are added. Makes a lot of sense. The second conformation we call open, and here there are certain characteristic movements. For example, look, TM6, it moves outward in a major way. There are other conformational rearrangements which correspond fairly well to what has been described in active conformations of the receptor. And you can see that this has opened up space on the inner surface of the receptor. And I think it's fair to say that this open conformation clearly corresponds to a classic active conformation of the receptor. If we come over here in light blue, you can see that there's almost no representation of that in the inactive forms of the receptor. The Berestin bias ligands, which are not able to activate G protein basically at all, again have very little. But once we get to angiotensin, we begin to pick up much more of this conformation. And finally, in the G bias ligands, the gain of function ones, they have more still. In fact, if we make a plot of the of that molecular efficacy parameter that we determined from the ligand binding before, that is log alpha GQ, versus the percent receptor population in this open conformation, you can see that reassuringly it provides a very good correlation. Finally, there are 2 other conformations which we refer to as occluded 1 and occluded 2. And we use this terminology because in each case, the inner surface of the receptor is partially occluded by one or another mechanism. In occluded 1, you can see that TM6, shown in green, doesn't move out in the classical way. And so it would be difficult for G protein to get in there. There's a partial occlusion of that inner surface. Similarly, for occluded 2, as shown here, TM6 does move out very well, almost like the open. But now another movement, intracellular loop 2, moves inward and blocks the internal surface of the receptor. Now, it's interesting, if we look here, it looks like these are the major conformations that are populated by Berestin bias ligands, and that's shown in brown and red. Interestingly, angiotensin itself also populates a significant amount of the occluded 2 conformation. So what the DEER study seemed to be showing is that the reason we can see biased agonism is not that biased agonists are able to populate unique, partially activated conformations, but rather that these conformations are also stabilized to some extent by the normal angiotensin analog. But what varies is that with the Berestin biased ligands, they pull the equilibrium away from the open conformation, which they don't populate, towards these occluded conformations. So they're somewhat promiscuous. By contrast, the GQ biased ligands seem to promote a very limited number of conformations, specifically the open or active one, thus leading to G protein bias. The other conclusion that comes from this is that the 2 transducers, beta-arrestin and the G protein, in this case GQ alpha, have very different specificities for interacting with these conformations. The beta-arrestins are, as I said, somewhat promiscuous, unable to populate a number of these different conformations, whereas the GQ protein is much more specific and appears to interact only with the open or classical active conformation. Now, having this information in hand is providing a boon to us in terms of trying to develop new biased ligands for a variety of types of receptors, and should ultimately be quite helpful in drug development. So in the closing minutes, though, I want to focus on one other, what I see as significant opportunity for drug development that comes from the recent biophysical studies of the receptor. So I've been talking up to now about biased drugs, and there are only, out of the 700 GPCR drugs, there are currently only 1 or 2 that are biased. There are also only 1 or 2 that are allosteric agents. Now, what do I mean by allosteric agents? Well, classically, as I say, you know, all but a couple of all GPCR drugs interact as agonists or antagonists with the so-called orthosteric site of the receptor. That's an outward-facing site, which is basically corresponds to the binding site where the normal physiological ligand binds. That's where adrenaline binds to an adrenergic receptor, where the beta blockers bind to adrenergic receptor, where histamine binds to its receptor, where antihistamines bind to the histamine receptor, etc. Allosteric ligands are ligands which bind anywhere else on the receptor, okay? And such ligands can either do nothing, or they can signal in their own right, or they can enhance signaling by a allosteric agonist, or they can diminish signaling by an allosteric agonist. If they enhance signaling by an orthosteric agonist, we call them PAMs, for positive allosteric modulators. If they diminish signaling by an orthosteric agonist, we call them NAMs, for negative allosteric modulators. These allosteric compounds have a number of very interesting properties which make them very interesting and potentially useful as drugs. And as I said, to date, just like with biased agonists, there are only 2 or 3 that are in use. All the others are orthosteric. So they're more selective. They have greater specificity than orthosteric drugs because allosteric binding sites are evolutionarily less conserved. There are more divergent sites and actions which can be targeted. There's only one small orthosteric site, but there are unknown numbers of allosteric sites located all over the receptor, which we're just discovering. So you can have multiple allosteric sites on a given receptor. There's greater on-target safety because allosteric modulators only affect the activity of receptors when endogenous agonists are present. Also, the effects of an allosteric modulator are saturable, so-called ceiling effects, which potentially reduce adverse effects caused by overdose. They also provide very useful tools for the study of receptors by biophysical techniques. Allosteric modulators are able to freeze or lock receptors into specific conformations by virtue of their cooperative interactions with orthosteric ligands. Well, why don't we have these drugs? Well, it really hasn't been possible until quite recently to find them. There was no way of looking for them. But over the last decade, we've pioneered methods in the lab, which we refer to as affinity-based selections for finding GPCR drugs. The idea is simple in concept, but not so simple in operation. The idea is you take a purified receptor, you immobilize it, say on beads, you then interact it with one or another huge self-encoding library. These could be nanobody phage displays, RNA optimers, or DNA-encoded small molecule libraries. We've used all three successfully, but we're most interested in the DNA-encoded small molecule libraries. These are huge libraries, 1 billion, that's with a B, 1 billion or more compounds, which each one having a small, let's say 80 nucleotide long tag on it, which serves as a barcode to identify each compound. And so the idea is you take your immobilized receptor, you interact it with the self-encoding library, you wash, you elute the small, tiny fraction of molecules which bind, you take those, you put it back on the column, and you cycle through repeatedly until you have enriched the binders sufficiently that they can be sequenced and characterized. One of the powerful things about this technology is that you can essentially load the deck. Let's say we're trying to find PAMs, positive allosteric modulators. Then we'll put an orthosteric agonist on the receptor. If we want to find NAMs, we'll either leave it empty or we'll put an antagonist on there. And then one basically uses deep sequencing to see what was enriched. One synthesizes those molecules and one tests them pharmacologically. And I'll just give you an example for the beta receptor, and we've done this with other receptors as well. We discovered a NAM when we compound 15, when we had an antagonist on the orthosteric site. What does a NAM look like? Well, here's a competition binding experiment looking at isoproterenol, inhibiting the binding of radioligand. As you add increasing concentrations of the NAM, you shift that curve to the right due to the negative allosteric effect of the compound, the NAM, on the orthosteric ligand. If you look at cyclic AMP generation, same thing, only here you're inhibiting the ability of isoproterenol to stimulate in a dose-response way cyclic AMP. If you look at Berestin recruitment, same thing. There is progressive inhibition of the ability of the orthosteric agonist to stimulate. Now, the opposite would be to have a PAM, and we have that. We call it compound 6, and here everything is reversed. It's the mirror image. In an isoproterenol radioligand binding curve, as we add compound 6, you shift the curve progressively to the left, in this case almost 50-fold. If you look at cyclic AMP generation, same thing. Starting with with no compound 6, you progressively increase the ability of isoproterenol to stimulate cyclic AMP. Same thing for beta-arrestin recruitment. So that's the classic behaviors of a NAM and a PAM. We even know where these molecules bind, which we had absolutely no idea when we carried out the initial screens. So these are crystal structures, which we obtained collaboratively with the Kobilka laboratory. For compound 15, the NAM, you see that it binds on the inner surface of the receptor, far removed from the orthosteric site shown here, occupied by the orthosteric antagonist carazolol, a conventional beta blocker. Here are views looking from inside the cell at the inner surface of the receptor. You can see the molecule basically snuggling into its little binding site there. And again, here, you can see that it makes interactions with numerous sites on the internal surface of the receptor, the internal surface of TM1, 2, 6, 7, and 8. So how does it work? Well, it has a dual mechanism, okay? First of all, it partially occludes the transducer binding site, and hence it's able to block the ability of either beta-arrestin or G protein to interact with the receptor. But it has another mechanism as well. And remember, I told you that transmembrane 6 is the most mobile of the inner surface. And if you look here, you can see in this animation that when you activate the receptor, TM6 normally moves out. But, and there are rearrangements, as you can see, in TM5, 7, 8, etc. But with compound 15 bound in that position, you can see that it's sort of like a spoke in the wheel. It's basically gluing a number of these helices together and preventing their free movement. So it's sort of a dual mechanism of action. What about compound 6, the PAM? Well, it also binds on the inner surface of the receptor, but in a different site. This one is framed by transmembranes 3, 4, and intracellular loop 2, which has this important short alpha helix. Again, you can see it bound there. Here's the same region of the receptor in green in the inactive receptor. So if I take the compound away, you can see that in the active receptor, there is this pocket into which the compound binds, but that pocket is constricted and unavailable in the inactive site. And so presumably the way we got this compound is since we had agonist on the orthosteric site, the receptor was spending enough time in this active conformation that this pocket was formed, and compound 6 could bind in there and stabilize it in its active conformation, and that thereby serves functionally as a PAM. So we believe that these type of allosteric agents, as with biased ligands, will in the next decade be increasingly found in clinical medicine. And I would say that one can also combine them. We have examples that I don't have time to talk about of biased allosteric agents, agents which can convert an orthosteric agonist like epinephrine or angiotensin, convert it into a biased ligand. So I think that is a coming wave in the future of therapeutics. Let me conclude by thanking some of the people currently in my laboratory for their work, also collaborators in the Kobilka and Hubble laboratories. Of course, I can't adequately acknowledge the dozens, even hundreds of people who've contributed in the work that I cited briefly earlier in the lecture, dating back half a century. Finally, let me shamelessly plug my memoir, which I published recently, called A Funny Thing Happened on the Way to Stockholm. I think this may be of interest to some of you. Not only can you find more about the scientific story, which is largely put into separate sections and chapter notes, but there are a lot of, I think, funny, amusing stories about my career path as a physician scientist, some advice, keys to success, and mentoring, etc. So I recommend that to you. It's available from Amazon or anywhere else that you buy your books. Okay. I'll stop there and thank you for your attention. And I will stop sharing that screen. You can see the book there over my right shoulder.
Hey, Dr. Lefkowitz, thank you so much for your time. It's a very fantastic talk. I think we're running out of time right now. We just want to thank you for your time and thank you for your wonderful contribution to the field. You not just GPR receptors, and in our cardiology division, we use these medications targeting GP cells almost every day. We just want to thank you again with all your contributions over the past 50 years. It's a very fantastic talk. Thank you so much. And for the
thank you very much.